flow through the ion channels. From the action potential in the cranial cavities. I'm doing it right. Sometimes some of those go through these. At the library. I think from me teaching you this because I think it's kind of fun to teach it. Let's get studying independent prep. I like to color code a lot. Oh my god, also I want to show you my little drawing. I know it's good. Own dry erase markers, and they also gave me theirs, but I might use mine. So these are my markers that I have. I'm excited because these are like really cool colors. And then the markers they gave me first of all I need this because I don't have one. Black, purple, and red. Looks like they will use my own and maybe the black one. So let's see. My my exam is tomorrow, so let's see how much I know. First, we're gonna do the pathway of the action potentials from the cranial nerve back to the brain and like where it goes. So the pathway to that. First, we'll do olfaction and then we'll do gustation. I'm gonna try to do this without memories. Also, don't mind the fit because I'm trying to be comfy, so that's why I'm wearing a really crusty outfit. I usually look good, I promise. Should be bigger. Take two. Also, if you couldn't tell, I'm playing Egyptian music because like dramatic instrumental music. Okay, so hopefully you can pick it up on the camera. Pathway of the olfaction from the action potential on the cranial nerve back to the brain. So I'm just gonna check. Okay, so this is what I have, and it seems right. So I first said the action potential from the cranial nerve 1, olfactory nerve, goes through the olfactory tract to the olfactory bulb, which is mainly just the areas in the brain. The common area in the brain is the piriform cortex of the temporal lobe, which then has an action potential go to the thalamus and the association areas. It could either do that, or it could go through the amygdala, where your memories and learning is associated with, and it goes through the hypothalamus. And this area is mainly with like smells that give us like emotional memory, so it gives us longer a visceral or emotional response to smells. Have the gustation pathway. I'm just gonna check. Awesome, I'm doing it, guys. I'm doing it right. Okay, so let me just walk you through it. For the action potential goes on cranial nerve 7, 9, and 10. Then it enters the gustatory nucleus of the medulla, which then goes to the thalamus, and then to the cortex, and then it goes to the different areas of the cortex. So it goes to insular taste cortex of the temporal lobe, it goes to operculum of the frontal lobe, and it goes to the amygdala and the hypothalamus. So taste associated to memories and emotion. I'm going to explain the odor and transduction pathway. You're going to use a black one because I don't know if you can see it on camera well. Marker 6. Did you guys just see that? I just actually fucked up. I used this like marker that I thought was an expo marker, and I used this on the board, and it was a permanent marker. And I got so scared because I'm like, I'm gonna show you guys this trick that I just learned in like 2 seconds. I wrote protein here. I wrote protein with this marker by accident instead of an expo marker, and it wasn't coming off. Look at that. Like, it's not coming off. This is coming off. This is coming off. This is it. So then I took this expo marker and I just did this. So make sure you just like scribble really nice with this expo marker. Just wait. I erased it and it's gone. I can't believe this. Like, are we seeing? I just discovered something and I kind of ate that up. Tag me when you guys use this hack, okay? Look, it looks good as new. Yay. Odin transduction pathway starts with the odin binding to the G protein coupled receptor, which activates the G protein receptor. That activates the adenylyl cyclase 3 protein, which increases the production of cyclic AMP. The increase in amount of AMP produced triggers and opens the ion channels. I spelled channels wrong, but opens the ion channels, letting the sodium and the calcium ions into the cell, which allows for depolarization to occur in the cell, and finally action potential on the olfactory bulb. Yeah. Now we're going to talk about gustation and its signal transduction pathway. So for gustation, there are 5 tastings, which are the sweet, sour, bitter, umami, and salty. Salty, sweet, sour, bitter, and umami. So 3 of those go through. Oh, I forgot it right now. But 3 of those go through metabotropic channels, and the other 2 go through ionotropic channels. So this is a good example of that. Metabotropic is using G protein coupling, and ionotropic is just ion channels directly associated with ion channels. Ions, yeah. So for sweet, umami, and bitter, I'll just make sure I'm right with this. First, the sweet, umami, bitter amino acids bind to the G protein coupled receptor. This activates the IP3, which is the second messenger in the pathway. Then that triggers the calcium channels to open, and the calcium channels are released. Then the vesicles are released, and naturally the neurotransmitters are released during the action potential. And then that allows for an action potential on the cranial nerves for gustation, which is 7, 9, and 10. Okay, and then for salty and sour, the pathway is pretty simple actually because it's ionotropic. Basically, the salty sour acids flow through the ion channels into the cell. Obviously, that creates depolarization in the cell and allows for calcium channels to open and calcium to be released. This allows for the vesicles to be released and neurotransmitter released, and then finally an action potential on cranial nerves 7, 9, and 10. Again, for gustation. I should probably review vesicle formation and reusing the pathway for reusing it. So yeah. Okay, this is exocytosis, calcium-mediated vesicle release, neurotransmitter release. So first, priming occurs, which is basically when the vesicle and the proteins are getting prepped and ready to dock. Next is docking, which is when I just realized. Okay, that's what I thought. Oh my god, I did not get enough sleep. So it's supposed to be. This is supposed to be right here. But this is 3, this is 4, and this is 5. Okay. Like I was saying, first priming occurs where the vesicles and the proteins associated with the vesicles are getting ready and prepping to dock. Then docking occurs, which the proteins synaptobrevin, SNAP25, and syntaxin wrap around each other and form the dock. SNAP25 is proteins on the vesicle membrane. SNAP25 and syntaxin are proteins on the plasma membrane, which basically they just turn around each other, twist, and wrap around each other to form like a snare complex. Then the syntagmin, which is another protein on the plasma membrane, binds to that snare complex, and then calcium enters from, the calcium channels open, calcium releases. Calcium enters and binds to the syntagmin protein, which has a calcium sensor, and calcium binds to syntagmin, and then that allows for conformational change, so protein shape change that makes the plasma membrane start to curve and bend around the vesicle. And then finally the two membranes, the plasma membrane and the vesicle membrane, fuse together, so exocytosis can occur, the neurotransmitters can release, and postsynaptic action potential can occur on the postsynaptic cell. Gosh, yeah, okay. Next, let's talk about vesicle reuse. Okay, here's a simplified version of recycling the vesicle membrane. So first, clathrin attaches to the vesicle during the initial budding of the vesicle and forms a cage-like structure. After the budding and the stalk occurs, dynamic membrane protein forms a little ring around the vesicle's budding and pinches it off for release. And then oxalin, which is just a protein, it breaks down the clathrin around the vesicle, so it could just be a plain empty vesicle. And then finally, the vesicle goes back into its normal shape and it goes back into the axon terminal. It goes back into the neuron terminal through endocytosis for reuse. It's 8:47, the library closes at 9, so they're gonna be kicking me out anytime soon if I don't leave. I'm going to continue studying for this class because my exam's tomorrow, and I'll let you guys know how it goes, keep you updated on my grade. So, yeah, thank you for being on this journey at the library, and I hope you learned anything from me teaching you this, because I think it's kind of fun to teach others. But yeah, thank you. The last thing I'm going to literally review with you guys, this is the last thing I'm going to review because I have 10 minutes. Okay, first, there's 3 receptors, acetylcholine, glutamate, and GABA. Acetylcholine is first formed at the neuron terminal, acetyl-CoA and choline, and those two form acetylcholine through choline acetyltransferase. The ACHAT loads the acetylcholine into the synaptic vesicles. Exocytosis happens, neurotransmitters are released. Acetylcholine is broken down into acetate and choline through acetylcholine esterase. And then the choline is then transported back into the neuron terminal through the CHAT, which is a co-transporter for reuse to make acetylcholine. Glutamate is formed with the enzyme glutaminase. So glutamine is converted to glutamate through glutaminase. And then glutamate is loaded into synaptic vesicles by V-glut. Exocytosis happens, neurotransmitters released, and then the glutamate is transported into the glia cell through EEAT co-transporter, and then that's where glutamate is broken down into glutamine, back into glutamine through glutamine synthetase, Then the glutamine is transported out of the glia through SN1 transporter and back into the neuron terminal through SMT2. And then the glutamine is reused to be converted to glutamine in the neuron terminal. For GABA formation, glutamine is converted into GABA through glutamic acid decarboxylase and pyridoxal phosphate to form GABA. Then GABA is loaded into the synaptic vesicles through VIAAT. Then exocytosis happens, GABA is released into the synapse, and GABA is transported into a glia cell. GABA is transported to the glia cell through the transporter GAT, which is where it breaks down into glutamate because of its concentration gradient and its breakdown. And then glutamate is then transported back into the neuron terminal for reuse to make GABA. In order for the GABA pathway to occur, vitamin B6 needs to be present. Deficiency in vitamin B6 will result in no GABA neurotransmitters being produced.